R.K Medicos. In this section, we are going to discuss about embryology part 3. And basically, in first part, we discuss about the gametes. In second part, we discuss about the first week of development. And now in third part, we are going to discuss about the basically second week of the development. And the second week of development is, it means second week, it means what? The day which start from the 8th day to 14th day. So we can see here the development from the what happened after 8th day of development till 14th day of the development. In first part we see about the gametes. In second part we see till this blastocyst stage, and they are going to implant. They are going to implant where in the functional layer, in the functional layer of endometrium. So this till this section we discuss in the second week of first week of our development. And in the second part of our embryology, first week of development of a human embryo, what happened with embryos? These are the blastocyst stage is going to implant into the functional layer of basically which endometrial wall. After that we are going to see what happened to these cells in the second week. What are the cells we see in that? That is the trophoblast cells and the embryoblast cells. Basically we can see these trophoblast cells are the outer layer cells. These are the outer, we can say like this, these are the outer cell mass, and the embryoblast is the basically inner cell mass. That these are the two cell masses, the outer and inner. The outer one is trophoblast. Tropho means nutrition, they provided nutrition to the developing embryo, and the embryo means proper embryo forming cells, and blastocell is the cavity inside it is formed. We already see about it. Now we are going to discuss about the part 3, basically development from the 8th day to 14th day, and that is the second week of development we can see here. And this is the picture we have to learn by heart from this, because this is the picture is whole scenario for the second week of development. And now we are going to see about it in the detail little bit. We can see, suppose that we can see like this. This is the this is our epiblast, and this is our hypoblast, and from the epiblast and hypoblast there is a special type of cells is forming on the epiblast, and they are the columnar in nature. These epiblast cells are columnar in nature, and this columnar in nature, columnar cells, and the hypoblast cells are basically, we can say like, these are the cuboidal. These are the cuboidal cells, and these both the cells, either hypoblast and epiblast cells, these are the cuboidal cells. We can say like this, these are the cuboidal cells, and these both the cells are forming from which layer? Inner cell mass. Inner cell mass. We are going to draw this picture from here. You can see this picture. This inner cell mass are the blue one which is present inside this region. This blue region cells, the inner cell mass are basically five cells in the number in the blastocyst stage. After that we are going to see how they are going to differentiate. They are going to differentiate in two layers, that is epiblast and hypoblast, and the epiblast layer is continuing. We are going to see in this picture, you can see these are the columnar cells. What I am going to paint by the yellow, these are the columnar cells is here, and these columnar cells are basically formed by the epiblast layer. And same we can see these are the hypoblast cells. These are the cuboidal in nature, and these both the cells are the differentiation of the inner cell mass which are present or inside the trophoblast or the outer cell layer. And later on we can say these epiblast cells or the columnar cells are going to forming some boundaries, or they are forming some membranous structure, and these structures are called as aminogenic cells. Aminogenic cells are produced from them. So we can say like this, they epiblast cells are forming a cavity and hypoblast cells are also forming a cavity. This epiblast is forming cavity above it, and the hypoblast forming cavity below it. This is the whole cavity which is formed by the hypoblast cells, and this cavity which form just below the hypoblast is called as primary yolk sac, and what the cavity is formed above the epiblast is called as what? Amniotic cavity. So now we are going to see what is the boundary of amniotic cavity is. The amniotic cavity is basically lined by the epiblast cells. This is the epiblastic cells layer through which these are lining. So I am going to delete it because we can see here these epiblast cells are going to form a boundary, and these hypoblast cells are going to also form a boundary. And these epiblast cells, what they forming a boundary is called as, there is also having some cells are present. We can see like this, and these cells are called as aminogenic cells. And here we can also see the hypoblast cells are forming that cavity, and that is encloses this cavity by the hypoblast cells. These cavities or these cells are also called as exocoelomic membrane. So these membranes are having, we can say like this, there is a flattened epithelial cells on this membrane, and these flattened epithelial cells are called as flattened epithelium or flattened squamous. Flattened means squamous, simple. So these are the flattened squamous epithelium. So till now we discussed only two things. What is the cavity in just above the epiblast is called as amniotic cavity, and what is the cavity just below the hypoblast? That is the yolk sac. And this cavity is basically called as, first time this cavity is formed below the yolk sac, so they are called as, suppose that they are primary, not suppose, these are basically, they are primary yolk sac, and above one is, that is amniotic cavity. Now we are going to see what happened to the trophoblast layer. The trophoblast layer is forming two different layers. These are the trophoblast layer here, one and another. So these two are trophoblast layer. From the initial, we are going to see only from the initial. We know from by heart there is a trophoblast layer and embryoblast layer. The embryoblast layers are the inner cell mass. The inner cell mass is basically divided into two parts, that is epiblast and hypoblast. The epiblast is enclosing a cavity, that one is called as amniotic cavity, and the hypoblast enclosing a cavity, that one is called as yolk sac cavity basically. And now the trophoblast layer is forming two different layers. The first inside layer is called as cytotrophoblast, and the outermost one is called as Syn Cytotrophoblast. So now you tell me, there is a Syn Cytotrophoblast, and cytotrophoblast is formed by the trophoblast, it is true. And what is formed by the inner cell mass or the embryoblast, they are the hypoblast and epiblast. And the hypoblast enclosing what? Hypoblast enclosing yolk sac or the primary yolk sac. Now we are going to see something important here. We can see there is a space present in between. Now imagine the space present in between the inner cell mass and the cytotrophoblast. The space, I'm going to mark here, the space which is present in between cytotrophoblast and the inner cell mass. It is true or not? Of course it is true, because the inner cell mass forming the two parts, that is the amniotic cavity and the primary yolk sac, and these two structures are combinedly forming the inner cell mass, or they are forming from the inner cell mass. So now you tell me these all structures are what they are representing here, a cavity, and this cavity is called as extra, we can see this cavity is called as extra embryonic mesoderm. Extra embryonic mesoderm. So the extra embryonic mesoderm is present where? It is present in between the inner cell mass. Inner cell mass is combinedly epiblast and hypoblast, and in between the cytotrophoblast of the trophoblast layer. So this is the extra embryonic mesoderm cavity, or it is the extra embryonic mesoderm part. Now you tell me the Syn Cytotrophoblast and cytotrophoblast, these two structures are formed on the 8th day of the development. On the 8th day of development of human fetus or the human embryo, development of human embryo. And in next phase we are going to see where the amniotic cavity is present, it is enclosed. Yolk sac is present, it is also enclosed. Now primary yolk sac is okay. So this is the structure we are going to basically discuss in detail about it. This is the development of second week of the human embryo. Now we are going to see in this picture. In Vivo View, we can see amniotic cavity is this one. This one is our amniotic cavity, what I am going to mark here. And this one is our yolk sac. We can see here, this is the yolk sac. This is the exocoelomic membrane. This one is exocoelomic membrane, the green part one, that is the exocoelomic membrane. This exocoelomic membrane is here, and this exocoelomic membrane is having flattened cells. Flattened cells you can imagine, and these flattened cells are, these flattened epithelial cells are lining this exocoelomic membrane. And what is this? This green color region is called as, this green color region is extra embryonic mesoderm. This is the extra embryonic mesoderm here. So hope it will be clear for you now. And now I am going to show you something. Now you can imagine where is the hypoblast, epiblast, and Syn Cytotrophoblast, where is the primary yolk sac. These are the structures we are going to discuss in detail in the second week of development. For now, hope this picture is clear for you. And now I am moving forward. In this picture you can also imagine, suppose that this is the endometrium of our uterus. We can see here this is the whole endometrium, and this blastocysts are implanted inside it, and they are going to further developed in the second week. And we can see this is the outermost layer is Syn Cyto, and just below it Cyto, and these are the combinedly to Trophoblast. These are combinedly to Trophoblast part of the embryo. Next we can see this is the which part? Epiblast, and this is Hypoblast. So we can see this is the Epiblast is here, and we can see this part is basically Hypoblast. This part is Hypoblast. And what is the cavity is his? This cavity is yolk sac. It is true. And this cavity is amniotic cavity. And next point we can see this is the extracoelomic membrane. We can see exocoelomic membrane is here, and they are lined by the flattened cell. Exocoelomic membrane lined by the flattened cell. You can see here exocoelomic membrane lined by the flattened cells. And what is this layer? This is the Syn Cytotrophoblast or cytotrophoblast. So this is the whole structure. Now you can imagine everything from the first structure. So hope it will be clear for you. In next video we can also see this. For now we can leave skipping. Next picture we are seeing later. Now you can read this and understand what we study above. We are going to study the inner cell mass is called as embryoblast. It is true. And the embryoblast is basically, or the inner cell mass is forming epiblast and hypoblast. These two cells are formed by the inner cells. We know by the heart from the initial. They are the columnar cells and this one is cuboidal cells. It is true. Next point we can see they are present in the dorsal aspects. They are present in dorsally, and hypoblast is present ventrally in the anterior part. Suppose that the someone is present like this anterior or the ventral part, and this one is the dorsal part at the ventral part, and this one is the dorsal part. The ventral part is participating in which hypoblast cells, and the dorsal part is participating in the epiblast cells. The epiblast cells in dorsal position. So what the epiblast is going to form later, now you know the epiblast cells is forming amniotic cavity, amnion, or they're forming the amniotic cavity, and these cells, these cavities are lined by which cells? Epiblastic cells, and these cells are called as amniogenic cells, or you can say like amniogenic cells. The amniogenic cells is lining the cavity, that is amniotic cavity. Same in the hypoblast, they are going to line a cavity with a membrane, that is exocoelomic membrane, and inside it there is a cavity is present. What the name of this cavity? The name of this cavity is called as yolk sac, and basically they are the primary yolk sac, and they are lined by a membrane, that is exocoelomic membrane, and the flattened squamous cells are present in that membrane. You know from the initial we can see these are the flattened squamous epithelium cells. These are the here we can see, and this is the lining, and this is the primary yolk sac structure, and this is the amniotic cavity, and this is the amniogenic cells here, and these are the hypoblast, epiblast. Hope it will be clear. Next point we can see in some patients. Now you can imagine there is a two round structures, or we can say like this, there is a two round structures you can see till this stage of the embryo development. Why we see two round structure? Because one structure is this one and one structure is this one. Suppose that we are going to view this whole structure, we are going to view whole this structure on the ultrasonography or the ultrasound or the ultrasonography. So what is happening here when we are going to see? So we are going to see two bubbles like a structure in this, and this bubbles like a structure is called as double blave sign. The double blave sign we are going to see on the sonography, and we can see here the double blave sign. And what is the double blave sign? Is it one is our amniotic cavity, that is rounded by the amniogenic cells, and one is our yolk sac, they are lined by the flattened cells or the exocoelomic membrane. So these are the two structures we can see on the ultrasonography, and they are looking like round, two round structure, the double bubble like bulb like structure. So they are called as two bubbles, or in this double blave shine, or the two bubbles like a structure is formed in the primary yolk sac, one is amniotic cavity. And when we are going to see these structures, we have only seen the second stage of, or the second week of the stage of development. We can also see here, you can see also, let me kill you. You can imagine now this is the one bulb and this is the another bulb, and it is inside the structure. Next point we are going to see, you can also read here, the double blave shine. Wait a minute, the double blave sign is sonographic feature where the visualization of a gestational sac which containing the yolk sac and the amniotic sac, giving an appearance of the two small bubbles like a structure. And that is also seen here, the embryonic one disc, that is amniotic sac and the yolk sac. So it is the picture view of our second week of development. Now we are going to see the outer cell mass, and the outer cell mass, you know now what is the another name of outer cell mass? That one is trophoblast, and these are providing nutrition to the cells. They are the outer cell mass, And the outer cell mass is having two parts, cytotrophoblast and Syn Cytotrophoblast. Now you know the cytotrophoblast is going to form something and Syn Cytotrophoblast is also going to form something. What is something is it here? The cytotrophoblast is forming the extra embryonic mesoderm. Now you can tell me the extra embryonic mesoderm is present in between what? It is present in between the inner cell mass. And inner cell mass, what is it? The inner cell mass is basically hypoblast and epiblast, and they are covering what is the amniotic cells and the exocelomic membrane. And the cytotrophoblast, this layer is present in between the inner cell mass and cytotrophoblast. We can see here once again in the previous picture, this is the extra embryonic mesoderm, and here is this, the extra embryonic mesoderm is it. It is present in between the inner cell mass and the what? Outer one structure is cytotrophoblast. So in between them, this extra embryonic mesoderm is present. Next, they are formed. What they are going to form? The first one, they are formed by this extra embryonic mesoderm is formed by three structures mainly. First one is yolk sac, is the first round structure. Next one is epiblast, the upper one which forming the yolk sac, and the hypoblast. These three structures are going to form basically in most of them, which are the structures which participate in the formation of extra embryonic mesoderm, which is the mostly formed. So the primary yolk sac, the above one. In the next section we are going to see Syn Cytotrophoblast. Syn Cytotrophoblast is outermost layer. Yeah, Syn Cytotrophoblast is basically going to form HCG. The Syn Cytotrophoblast is secreting something, that one is called as HCG, human chorionic gonadotrophin hormone. This hormone is secreted by the Syn Cytotrophoblast, and it is the outermost covering of our what? Embryonic development. And these are all going to basically embedded inside the endometrium functional layer, and they moving inside it, and they going to secrete human chorionic gonadotrophin hormone. This HCG is secreting by them, and this HCG leads to the secretion of progesterone from it. This HCG is also providing, or they are going to release what? The Syn Cytotrophoblast is going to promote the HCG to release the progesterone for the maintaining of pregnancy. The maintaining of pregnancy is done by which hormone? Progesterone. And the progesterone is secreted by which hormone? That is HCG. In initial stages, in the primary stage or the early stage of our growth, the HCG is secreting progesterone, and that progesterone leads to the maintaining of our pregnancy. In next point we can see what is the assay of HCG. HCG basically how we can determine it. HCG on the 8th day we can check. On the 8th day of the development of the human embryo, we are going to check the maternal blood, and we find the HCG, and we conclude that the lady is pregnant. In next case, on the 9th or 10th day of the development of any female, we are going to check the maternal urine, and that is in which the concentration of HCG will be found, and it's also going to say that there is a pregnancy. And on the first day, suppose that the any lady which is having absence of the menstrual cycle, so he or she goes to basically she goes to for the first day of the missed period, and check by the maternal blood, the HCG level inside it, and she is going to identify for the pregnancy, if she is pregnant or not. Next point we can see, when the HCG level will be low, the HCG level will basically low at the time of when the maybe lady have ectopic pregnancy. And what does it mean? Ectopic pregnancy is simple, the fertilization in fallopian tube. Suppose that the fertilization which happened in fallopian tube, we already discussed in the second part, so you can go and watch. This fallopian tube fertilization is leads to, suppose that their tube diameter will be smaller, they are not coming into the uterine cavities, so these uterine, inside the uterine tube, they are going to they are going for the growth. And this Syn Cytotrophoblast, what is the outermost layer of this embryo, developing embryo, they are not going to develop that much, so their secretion will be very low and they are not going to produce that much HCG. The HCG is not produced by them, so it will be in ectopic pregnancy, the HCG level will be also down, because Syn Cytotrophoblast is not that much developed, or they are the poorly developed. Next point, we can see there is a spontaneous abortion. There is any ladies having abortion spontaneously, so that is also leads to the less amount of HCG in the blood. Next point, we can see in what condition the HCG level will be higher. The HCG level will be elevated in two cases. First, if the patient or if the lady is having multiple pregnancy, maybe they are having multiple pregnancy or the molar pregnancy. What does this mean, molar pregnancy? Multiple, you understand, but molar is the abnormal growth of placenta or the abnormal placenta with proliferation of chronic villi. Chronic villi we discussed just after that, so I'm not going in detail about it, but we have to understand it. There is a abnormal placenta with proliferation of chronic villi. In these cases, what happens, there is two types of molar pregnancy. We can see there is a partial mole or the complete mole. What does it mean, partial mole? The fetal parts are only few parts of our fetus we can see, and that fetal parts is leads to the pregnant. We can see in the molar pregnancy basically only few parts of the fetus we can see, but in the complete mole the fetal parts will be not present there. There is no any fetal parts, there is only big placental parts is seen in the cluster form. We can see the complete molar pregnancy, there is only fetal part is not present, only placental part is present there. But in the partial mole there is some parts of the fetus is present and the placenta is remaining part is there. We can see here the fetus, some normal villi are present, but the fetus villus hydrops, but the gross fetal parts are also present here. But here fetal parts are not present, only placental parts are present. So this type of pregnancy is called as molar pregnancy, and you can also see in the picture the partial and complete. In the partial you are going to see this fetal parts, but here no any fetal parts is appear in the complete molar pregnancy. In next part we are going to discuss about the function of chorion and primary chorionic villi. What is the chorion and how they developed, and what is the chorionic villi is it? Now you tell me. I'm already told you that these two structures are called as trophoblast. From the first picture, this trophoblast, the outermost layer is Syn Cytotrophoblast, and just below it this one is called as Cytotrophoblast. And this Syn Cytotrophoblast is going to form something, and Cytotrophoblast is forming the primary chorionic villi. How? We discuss later. In next section we can see here, this is the epiblast and this is the amniotic cavity, this is the yolk sac, this is the exocoelomic membrane, and exocoelomic membrane is lined by flattened cells. This type of structure is also present here. After that, now we are moving up to the next level. We can see the primary villi is formed by Cytotrophoblast and Syn Cytotrophoblast. Now you told me this is a Cytotrophoblast and this is the Syn Cytotrophoblast. Some cells from the Cytotrophoblast, this is the Cytotrophoblast, some cells are going to embed in the Syn Cytotrophoblast. Some cells from the Cytotrophoblast is going to embed in the Cytotrophoblast, and they are forming the primary chorionic villi. The chorionic villi or the primary or the first one is formed by basically the proliferation or the embedding of the Cytotrophoblast cells are embedding into the Syn Cytotrophoblast cells. So these are the Cytotrophoblast cells layer, and these cells are going to embedding into the Syn Cytotrophoblast, and they forming what? Primary chorionic villi. and these villi, finger-like projections, are formed embedding inside the Syn Cytotrophoblast. Before the 12 days they get formed. Here in next step we are going to see, wait a minute. Now next point is Syn Cytotrophoblast. Now you can see the Syn Cytotrophoblast is invading this Syn Cytotrophoblast. Now we can see here this Syn Cytotrophoblast cells. This all layer is Syn Cytotrophoblast. Now this Syn Cytotrophoblast is embedding in, this is the outermost layer, Syn Cytotrophoblast. Basically I am taking from the initial, this Syn Cytotrophoblast is the outermost layer, and these layers are embedding inside the endometrium. Because these layers are the outermost and they are embedding endometrial walls of the uterus, these are also embedding the endometrial vessels. Why they are embedding endometrial vessels? Now you know there is in the endometrium during the implantation or after the during the development time, the endometrial glands and the endometrial blood vessels are gets proliferated in the functionalis layer, and in that whole the uterine cavity, the endometrial layers is full of blood vessels. They are full of endometrium, is having endometrial glands, and this Syn Cytotrophoblast layer of this embryo are going to embedded in these blood vessels. They are going to embedded in the endometrial walls, and what is happening next? They gets embedded in the endometrium, they gets in the endometrial blood vessels, and they forming some round lacunas like structures. Lacuna means basically vacuum, or we can say like this, the round structures with field of blood, and this is the forming lacuna with maternal blood. There is a, inside these lacunas there is a maternal bloods are filled. Lacunas means the cavity which filled of blood. We can see here these are the lacunas, and these are present where in the Syn Cytotrophoblast layer, and they are going to nourish. In next step we can see here these maternal bloods what are filled in this lacuna are diffused into the inner cell layer. What these layers are filled with blood, they are moving towards the inner cell mass, and inner cell mass is basically two parts. Now you told me there is a two parts, epiblast and hypoblast. Epiblast plus hypoblast. And what contains the cavity is form encloses the epiblast is amniotic cavity, and hypoblast is yolk sac. So basically this lacuna is filled of blood and it is present in Syn Cytotrophoblast layer. They are going to nourish, or they are going to blood supply to which layer? Inner cell mass layer, and inner cell mass layer is basically epiblast and hypoblast part, and this amniotic cavity and yolk sac part is going to nourished by them. So now you are going to say what is the Syn Cytotrophoblast blood vessels or the field of lacuna. They are transferring their blood into the inner cell mass or the hypoblast or epiblast layer, and this is called a uteroplacental circulation. What is the transmission of these bloods from the Syn Cytotrophoblast to the inner cell mass layer? This is called as uteroplacental, because these bloods are coming from the uterine wall through these placental targeting parts, and these are called as Syn Cyto. We can say the uteroplacental circulation. This is happen on the 15th day of the development. On the 15th day of development, uteroplacental circulation is established, and this uteroplacental circulation is supplied by lacuna of the maternal blood. These are supplied by the maternal blood cells. Till now the child or the embryo have no heart, no blood system is formed, no cardiovascular system is formed. So there is a blood supply is coming from where? The blood supply is coming from the mother, and these lacuna are filled of blood and they supply to the child through the which type of circulation? Uteroplacental circulation. Now you can see here how. What I told you, these are the lacunas. We can see here these are the lacunas which are present in the Syn Cytotrophoblast. green one is Syn Cytotrophoblast. These are present. These are the lacunas, and inside it's the red color is filled of blood. These red color inside it, suppose that this is the lacuna, this is the field of blood, and from here they transferring the blood to the, they are going to, these are the lacunas, they are filled of blood, they are going to transfer their blood to the inner cell mass. Once again, in the epiblastic layer also they are filled of blood, they are going to transfer their blood to the inner cell mass. These are the lacunas, they are transferring their blood to the inner cell mass. In this manner they are going to nourishing the amniotic cavity or the yolk sac, or we are going to, or you can also say like this, they are in the epiblast layer or the hypoblast layer, they are nourishment, they are providing nourishment to the embryoblast cells. And this circulation is called as utero-placental circulation, because these bloods are coming from the maternal source or maternal uterine walls. That's why they are called as utero-placental circulation. Hope you understand now, because these are the circulation, these bloods are providing them, they are moving like this and then nourishing the inner cell mass. Next we are going to see, when the first time the first organ system is developed in the fetus. What is the first system is in any child or the any developing embryo is formed? These are the cardiovascular system. Basically the cardiovascular system is the first system which is formed or developed inside the developing embryo, and along with the cardiovascular system there is a blood vessels are also formed, and the heart is not totally formed, the heart tubes are formed. There is the formation of heart tubes in the first developing system, and after the formation of heart tubes and the cardiovascular system, there is a feto and placental circulation is established on the 22nd day. This circulation, which one? The feto and placental, between the placenta, through the placenta, and the fetus is gets taking the blood from his heart and they transferring from the mother to child through it, and this is the feto. Basically this feto-placental circulation is established on the 22 days, and in some books there is also written the 17 days, so you have to memorize that is basically 22nd days for now. And these are the first organ system which is developed in the developing embryo in the second week of the development, or in some books they are also written like the 17 days. Now we are going to see the secondary chorionic villi. The primary is developed on before the 12 days. They are synthesized from the cytotrophoblast and Syn Cytotrophoblast. The cytotrophoblast cells are going to embedded in the Syn Cytotrophoblast finger-like projection, and they are forming the primary villi. Now we are going to see the secondary chorionic villi, how they are formed. You can see here in this picture, this is the yolk sac, this is the amniotic cavity, and this is the epiblast, hypoblast, epiblast, and now you tell me, this is the outermost layer is called as Syn Cytotrophoblast, and next this layer is called as Cytotrophoblast. And what is the cavity which is present in between the cytotrophoblast and inner cell mass? That one is called as, this whole cavity is called as extra embryonic mesoderm, EEM, that is the extra embryonic mesoderm. This is the cavity, and inside this cavity there is a something is formed. We can see here before going to detail. Now you tell me once again, we are going to see in this picture week 2 development here. This one is Syn Cytotrophoblast, or we can say like this, this is the whole structure is extra embryonic mesoderm, and inside the extra embryonic mesoderm there is a special type of cavity is formed. We can see here something, and just below this, just inner to it there is a red structure, and just outer to it there is a red structure. Some name of this and some name of this. What is it? We are going to see now. We can see in this picture. I am going to tell you the outermost layer. Now you tell me. The outermost layer is Syn Cytotrophoblast. It is true. Just below it, Cytotrophoblast. Next point. This cavity is yolk sac and this cavity is amniotic cavity. It means just below it, epiblast and hypoblast. After that we can see this whole region. This whole region is what? Extra embryonic mesoderm. Extra embryonic mesoderm. This whole region is extra embryonic mesoderm. And in this whole region you see one cavity and one membrane is here and one membrane is here. What is this? This is the basically separation of extra embryonic mesoderm into two parts. The inner one is called as this inner structure. This inner structure is called as, we can see here, this inner structure is called as a splanchopleuric layer of, we can see here, a splanchopleuric layer of extra embryonic mesoderm. And what is this one is called as? This one is called as somatic or parietal layer of extra embryonic mesoderm. So these are the three parts. There is a one cavity in the extra embryonic mesoderm. There is a one cavity inside it and the two parts. That is the extra embryonic mesoderm is having three parts, one cavity, and the innermost part is called as a splanchopleuric layer of the extra embryonic mesoderm, and the outer one is called as a splanchopleuric layer of extra embryonic mesoderm. So now we are going to see these all structures what we discuss now. Wait a second. See here, this extra embryonic splanchnic mesoderm is also called as a splanchopleuric layer of extra embryonic mesoderm, and here is this layer. We can see this structure is representing this whole round structure. What I am drawing here, this structure is representing here what? This structure is representing here inner part of the extra embryonic mesoderm is going to represent extra embryonic splanchopleuric layer of or splanchnic layer of the mesoderm. These are the innermost part. In next we are going to see the outermost layer, this one is called as, we can see here, this one layer is called as parietal or somatic mesoderm. This layer is called as parietal or somatic mesoderm. Parietal or somatic mesoderm of extra embryonic layer. So hope it will be clear for you now. Let me clear everything. We will discuss once again. Let's start. Let me draw for you. This one is epiblast, this one is hypoblast, this one is yolk sac, this one is amniotic cavity, this one is yolk sac, and this one is cytotrophoblast, and here is the Syn Cytotrophoblast, and this one is extra embryonic mesoderm. It is present in between the inner cell mass, and this one is cytotrophoblast. Hope now it will be clear. Cytotrophoblast. and this extra embryonic mesoderm is having one cavity is formed inside it. This extra embryonic mesoderm, this is the extra embryonic mesoderm, this is the extra embryonic mesoderm, this is the extra embryonic mesoderm, and it is present in between the inner cell mass and cytotrophoblast. Okay, now this inner cell mass or cytotrophoblast layer, and between them there is extra embryonic mesoderm. This is forming two parts, one structure, one layer inside it throughout the which part, exocoelomic mesoderm. Throughout the exocoelomic membrane they are forming a one layer, and one layer with the cytotrophoblast, and in between them there is a cavity is present. We can see here. And here is this cavity. You can see now. And this cavity is present where? In the chorionicvilli. Now you know chorionicvilli is also going like this. So this is called as chorionic cavity, basically extra embryonic chorion. This is called extra embryonic coelom cavities. These are the chorionic cavity. So hope it will be clear for you now where is the extra embryonic membrane is located, and where is the cytotrophoblast, Syn Cytotrophoblast, yolk sac, amniotic cavities, and everything. Hope it will be clear for you. Now you can look at it and watch and understand what is happening here. Now this one is clear, extra embryonic mesoderm, parietal or somatic, that is the outermost layer of the extra embryonic mesoderm. This is the splanchnopleuric layer, this is the cytotrophoblast, the second most, and the Syn Cytotrophoblast, the outermost. And now just imagine the Syn Cytotrophoblast, wait a second, Syn Cytotrophoblast, and the extra embryonic mesoderm from outside, that is the somatic or pleuric layer. These all three together are called as secondary mesoderm, secondary chorionicvilli. These all three together are called as secondary chorionicvilli. What is the primary chorionicvilli? The primary chorionicvilli is going to intervading the cytotrophoblast and the Syn Cytotrophoblast, but these three are combined together. That is the extra embryonic mesoderm. Which part of extra embryonic mesoderm? That is the outermost part. And outermost part, what is it? That one is the somatic or parietal layer of mesoderm. This somatic and parietal layer of mesoderm is combined with Syn Cyto and cytotrophoblast and forming this structure. You can see now this one is the whole structure which is representing here. This whole structure is representing the secondary chorionicvilli, and in this structure now you can look, this structure, and this structure, these three are adding together and they forming the secondary chorionicvilli. So it means cytotrophoblast, Syn Cytotrophoblast, and the extra embryonic mesoderm. Which part of extra embryonic mesoderm? That is the somatic or parietal part. So hope it will be clear for now. Next, wait a second. In next part we can see the cavity inside the extra embryonic mesoderm. Now imagine the extra embryonic mesoderm, inside it the cavity is present. It divides the mesoderm, divides the extra embryonic mesoderm into two parts. That is the parietal layer, that is the outermost, and the innermost is splanchnopleuric layer. And this splanchnopleuric layer is the innermost and outermost is parietal layer. Now the amniotic cavity, where it is located? The amniotic cavity is located just above the epiblast or hypoblast, and it is forming by the membrane that is enclosing a cavity. That one is called as amniotic cavity. Now imagine this amniotic cavity is lining externally by the something and internally by the something. Internal wall of the amniotic cavity is lined by the aminogenic cells. Now you know these aminogenic cells or the aminogenic cells are the lining internally. These aminogenic cells are going to line internally what amniotic cavity. But what is lining the externally? You can see here. This is the lining of internal amniotic cavity, and now you know these cells are what? Amniogenic cells from the first diagram. And there is also something is present which covering it from here. What is this? This layer is till here. Till here. This all the structure. What is it? This one is the splanchopluidic layer of mesoderm, a splanchopluidic layer of external mesoderm. But this layer, this all layer together, this layer and this layer, these all together are combinedly called as somatic or pluidic. It means the somatic and parietal layer, somatic and parietal layer of the external or the extra embryonic mesoderm are going to line this cavity. This cavity is lined externally by this extra embryonic mesoderm, and that extra embryonic mesoderm is parietal or somatic. The cavity, or which cavity? The amniotic cavity. The amniotic cavity is lined by the above or externally through the one and only extra embryonic mesoderm. And what is the extra embryonic mesoderm here? Now you know what is it. I'm not going to tell because it will take a lot of time to tell me. Next point, you know now here, this externally and internally, the parietal layers of the extra embryonic mesoderm is covering, and the internally the aminogenic cells. Now the secondary chorionic villi, you know how it is formed. The secondary chorionic villi is combination of three structures, that is somatic or parietal extra embryonic mesoderm plus cytotrophoblast plus Syn Cytotrophoblast, and these all three structures are combinedly called as secondary chorionic villi. And what is the primary chorionic villi? There is a combination of cyto and Syn Cyto, so these are called as primary chorionic villi. They are formed on the 12th day of development. Which one? The secondary chorionic villi is forming on the 12th day of development, and the primary chorionic villi is formed before the 12th day of development. So you have to memorize it. In next section, now you say note about the some important points. Before we are going to tell, we can see here summary. The two layers of inner cell mass. What is it? Epiblast and hypoblast. Inner cell mass having two layers. Now the two layers of outer cell mass, you can tell me what is the two layers of outer cell mass? That is cytotrophoblast and Syn Cytotrophoblast. That is cyto and Syn Cyto. What is the two layers of extra embryonic mesoderm? The outer and inner, the splanchopluidic layer and the parietal or somatic layer. Next one, what is the two villi? Do you know? That is primary villi and secondary chorionic villi. And what is the two layers are formed? Amnion and choriones are formed here. And what is this? Connecting stalk is here and umbilicus. These are the undivided part of the extra embryonic mesoderm. And where it is located, you can see here. This is the region where extra embryonic mesoderm are not going to divide. This region is extra embryonic mesoderm is not going to divide, and this region is called as primary stalk. And we can also see here this layer is called as connecting stalk also. This layer is also called as connecting stalk. In this layer, the extra embryonic mesoderm are not going to divide properly. So this part is connected with cytotrophoblast, and these are helpful in the formation of placenta or the umbilicus later on. So this is the picture. Now you are just going to take a review for 2 or 3 minutes. You can see here, this is the amniotic cavity. You can see this is the amniotic cavity. This is the primary yolk sac, and this one is extra embryonic mesoderm. This whole layer is extra embryonic mesoderm. This is Syn Cyto and cytotrophoblast layer. This is the cavity is extra embryonic xylem, and we can see once again the extra embryonic xylem is here, and their innermost layer is splanchnopeluric. The outermost layer is parietal or somatic, and this one is cytotrophoblast, and this is the aminogenic cells. This is the amniotic cavity, yolk sac. This is the hypoblast. This is the epiblast. These are the columnar cells. These are the cuboidal cells. Once again you can see these are the primitive or the primary yolk sac. These are the amniotic cavity. These are the epiblast, hypoblast, and the extra embryonic splanchnopeluric layer. This one is splanchnopeluric layer, and the outer one is splanchnosomatopeluric layer of that, and we can see cytotrophoblast cells, the outer one, and these are the cavity inside the extra embryonic mesoderm is extra embryonic xylem. Next there is two villus, primary and secondary villus. Do you know primary and secondary villus? Primary villus is formed, primary chorionicvilli is formed before 12 days, and secondary chorionicvilli is formed on the 12th day. And all the 12 day they are formed by the three different parts, that is extra embryonic mesoderm, cytotrophoblast and Syn Cytotrophoblast. These are the main regions, and you know the primary villus is formed by these cells of the cytotrophoblast are going to embedding inside the Syn Cytotrophoblast. These cytotrophoblast are going to embedded in Syn Cytotrophoblast, and they forming the primary villi. After that they are on the periphery region, and they are forming secondary villi. So this is all about the second week of development here. Hope you understand everything clearly, and in next section we are going to discuss about the third week of the development in the fourth part of the embryology. Thank you so much. See you in next video. Bye bye.